My name is Mike Prerau. I'm a researcher here at Brigham and Women's Hospital and Harvard Medical School. And today I'm going to talk about some of the work that my lab's been doing over the past couple of years. In particular, an open source software package called DYNAM-O, the Dynamic Oscillation Toolbox. And it's based off of the paper that you see here below, Stokes et al. From this past year. And so everything that we have here, we have full tutorials, code, all of our GitHub is available at our website, sleepeeg.org, and we welcome you to take a look at these. And we have this is for both MATLAB and Python. So let's get started. So what on earth is a sleep spindle? And in essence, a sleep spindle is a specific time domain waveform that an expert says is a sleep spindle. Now, we have some sort of general understanding and qualitative guidelines that have been developed for what a spindle is. But, these are typically these events, football-like events that occur at about 2.5 to 3 spindles per minute, at least classically. And we have learned through a lot of rodent work that from a neurophysiological standpoint, the activity of spindles is linked with thalamocortical networks, generated in the TRN associated with hippocampal ripples and thalamocortical upstates and a bunch of other things, right? And why are spindles interesting to us as researchers? Obviously, there's the relationship between spindle activity and memory consolidation. There's the relationship between spindle deficits and various psychiatric and neurodegenerative disorders. And there's, of course, age-related changes, which gets people interested in terms of biomarkers of dementia. And you see from two publications here, the change in spindle activity over the course of the lifespan, and then the change in spindle timing with respect to the slow oscillation with aging. And so that's made them, of course, a very interesting topic of inquiry. But where did we get spindles from? Well, in the 1930s, Alfred Loomis and colleagues looked in EEG recordings and found this first report in 1935 and sort of described them as these regular bursts at 14 hertz. And we've designated them as spindles because that's how they look. And they also found these things they called balls and also 5 other types of spindles that they can distinguish that they didn't really talk about. But really, what they were doing is trying to look at these EEG recordings made from these pens that were moving back and forth very slowly and had essentially a bandpass filter limiting what they're doing. And what most people do clinically for spindles is try to identify them in the time domain, looking in at the waveform traces. And this is classically a very difficult problem. So what we have here is a simulation here where we have some sort of low frequency, slow oscillation activity, some 1/f noise that keeps varying, and various spindle-like waveforms that move across. And as you can see, depending on where these spindles are, how much noise there are, where they appear relative to slow oscillations, they can be very, very difficult to see. Finding these types of waveforms within actual EEG is a very, very difficult problem. And that's borne out when you look at spindle identification by experts. You can see that this is from the DREAMS database. We have a bunch of different records that were scored by 2 different experts, and the numbers are vastly off. And in case you think expert number 2, which is the greedy spindle detector, here's another record in which the expert number 1, which scores lower on the other ones, is twice as many. So it's difficult. Now, this has led itself into differences between human scorers, automated algorithms, and of course, whatever machine learning approach you have, if it's based on a ground truth made by either human scorers or automated algorithms, what this is doing is it's imposing a potentially false rarity assumption on the nature of these events. And so you have these circular definitions that a spindle is what I see, that I'm going to detect something based off of criterion, based on what I see, and then it sort of feeds forward. So, maybe these underlying assumptions are incorrect. So how are we going to address these? Well, obviously looking at these in the time domain has the problems that we observe, but if we look at the same simulation in a spectrogram in the time frequency domain, you can see that the spindle activity appears as these very clear blobs. And really, no matter how much noise you throw out these things, you can really see that. And this is based off of the general principle that you get at when you look at various forms of Fourier analysis and its derivatives, is that transient oscillation, so an oscillation that's spindle-like, we'll call it transient oscillation, when you see it in the time domain, will be a peak in the spectrogram. We call these time frequency peaks or TF peaks, which we'll refer to moving forward. So as you look in an actual EEG spectrogram for a period of about an hour of non-REM, and then progressively zoom in on the sigma range, you can see that we have all of these blobs here, these time frequency peaks that correspond with no spindles. And so we published a work a couple of years ago, basically demonstrating that, in no other way besides amplitude difference do these time frequency peaks differ from traditional spindles. And that makes sense, right? Because we're looking at things historically in the time domain, you can only pick out the sort of the biggest, juiciest spindles that occur at the periods of the lowest noise. And so that's essentially how our assumptions about spindle activity exist from what people could see by eye in records from the 1930s and beyond. So what do we do in this case? Well, instead of trying to detect these, we try to detect these time frequency peaks. And we use a variant on what's called the watershed algorithm, which was a computer vision approach. But essentially, we look at the spectrogram as a 3D topography, and we're able to extract these. These are actual spindles here, spindle peaks here. So what we also try not to do is assume what is and what is not a spindle, because we just don't know. And rather than doing that, we look at these as a distribution. So what we can do is we can take an entire night spectrogram, a high resolution spectrogram, and from that identify 30 to 60,000 peaks a night. And instead of deciding, well, what is and what is not a spindle or a time frequency peak, we just look at the distribution of these. So this is the average rate or density of these across the entire night. And when we looked at this was really interesting across subjects. In a cohort of very well triaged, healthy subjects, we saw a lot of differences. We had, some subjects with 1 bumps, 2 bumps, maybe fast and slow spindles that we know about. Then we saw bumps in the low alpha range, along with some traditional spindle range activity. And then we saw some with 3 and even more. And just the variation was incredible for this actual small group of very, very well triaged, healthy, normal subjects. So what you can see is that these are heterogeneous. And one of the things I didn't say is that we had 2 nights here. And if you look at these actual density plots, behind them is a gray line, which you can almost not see, from a second recording that took place 1 or 2 nights later. And these are just bang on. So these are trait-like across nights, but very heterogeneous across subjects. And so what we can do, instead of taking the aggregate across all sleep, well, we can look at these density of these TF peaks as a function of frequency across the different stages, and we can turn those into sort of a color plot here, where you can see with an increasing depth of sleep, we can see what this is the density now that we're looking at of how many events per minute per bin we observe. And so this gives us this general picture of what's going on. But of course, sleep is a continuous dynamic process, and sleep staging is not only subjective and highly variable, but it discretizes sleep. It discretizes sleep in terms of a number of fixed phase states, so fixed 5 stages of sleep plus wake. And then it also discretizes in time. Well, you're contained to 30-second epochs, right? So we really have to say that in going to discrete stages, we're losing both state and temporal dynamics. So what do we do? Well, instead of sleep staging, we look at slow oscillation power, which has long been sort of known as a continuous correlate of depth of sleep. If you look into the 2-process model and the hypnogenic process S, and what you can see here is this is the slow oscillation power, which in this case is from 0.3 to 1.5 hertz extracted. It's a beautiful mirror image of the scored hypnogram below. And so here's what we can do. We take a look, here's a spectrogram, and here are all the TF peaks that we extracted over here. And here's the slow oscillation power. And what you can see here is this is the hypnogram itself mirrored upside down on top of it, so you can see how well that tracks. So here's what we do. Rather than just looking at REM 1, stage 1 through 3, non-REM stage 1 through 3, what we'll do is we'll look at a window of slow oscillation power, with the lower slow oscillation power representing a lighter state of sleep. So we look at that level of slow oscillation power, and we look at all the time frequency peaks that occur during that time, and we look at the density of that. And we can slide that up and up and up and up to make a continuous version of this. So when we change from discrete stages to continuous depth of sleep, it's like putting on glasses. We now see that there are two clear modes of activity transitioning from this sort of fast spindle mode to a slow spindle mode as you go continuously into depth of sleep. And so this matches with our original phase, a sense of where the stages are. You can do the same thing with time frequency peak phase. It's well noted that spindles tend to couple with slow oscillation activity, which we can sort of model as a sinusoid and look at where in the phase of this sinusoid or this wave does this occur. And this, of course, is important from what I discussed before, that as people age, you can see a shifting towards the rising phase of the slow oscillation with age. So this is interesting. So what do? We look at all these time frequency peaks here. We compare them to the slow oscillation and figure out what their phase is. So what do we do now? We can take this, this scatter plot of time frequency peaks here and now color it based on the phase. And what you can see here without doing anything is we have sort of a band of sort of light green here and a band of dark blue. And so what we observe here is that we can actually look and look across phase here and characterize it in the same way that we have these occurring at these different phases, and then slide up just in the same way and show that at what phase is what proportion of the events at each frequency correspond to the phase that we have. Okay, so we can put this all together and we see that these are a robust EEG fingerprint, that we see this again, these are healthy subjects, but night to night stability, but within subject, high heterogeneity. And this, as many subjects as you have, you see these vast differences. And this persists. This is from the SHHS, where you had 2 visits 5 years apart. You still see that there are some differences, but overall the structure maintains a great similarity. And we can look at this from the Cleveland's family study and look at there are these robust changes across the lifespan where you see these modes rising in some of these lower modes. There are clinical applications for this as well. Our paper looked at differences in activity in time frequency peaks for patients with schizophrenia. If we look at the average of these, we call these slow oscillation power histograms for healthy controls versus patients with schizophrenia, we see a marked difference. And in looking at this, we sort of found that there's a known fast spindle deficit, but we were able to find novel deficits in low alpha time frequency peaks and theta that could suggest sort of different potential new avenues for clinical applications. And so overall, what this allows us to do is, in a very principled way, understand the phenomena, allow us to observe things that are occurring that are spindle-like at all frequencies and times of nights on a continuous way. These are really robust night to night and month to month, and in some cases year to year, but the variability of that might tell us something clinically. And so this allows us to look on a continuous standpoint, continuous scale of sleep and compare across subjects. So, and so we will use this and are using this as a biomarker framework for detection and description of neurodevelopmental age. So just wanted to finish up saying, again, you can find our code and tutorials at sleepeeg.org. We have in-depth tutorials walking you through step-by-step how to create these and what to do with them. And you can basically go to our website, click on this this DYNAM-O page, and then over here you can see this is our sort of our tutorial that we have here that will sort of walk you through some of the background and then start getting into some of the example scripts and functions. So thank you all so much for this. And I just wanted to thank a bunch of the members of my lab and collaborators. And if any of you are interested in reaching out, my number is my email is right over here, and we can talk about any help you need and other potential collaborations. So thank you so much. So there's a question about, do time frequency make sense for high frequency events, but they don't make sense for low frequency events? It's not transient, it's not a peak. Well, it depends. So it depends how low frequency you're talking about. We usually stop at about 4 hertz. So essentially, you can understand whether or not something is some sort of some sort of state switching process versus a continuous oscillation. And there are other techniques to that you can use in some of these new state space models that are coming out that sort of allow you to assess whether something is a transient or a continuous oscillation. But again, it depends what you determine. A peak can be as long as you want it. It can just be a very, very long peak, right? And so I think there's sort of a balance of these things, but at a certain point, the frequency gets low enough such that you cannot actually discern the difference between that from spectral bleeding from the slow oscillation. All right, excellent. We have one more. Can your approach be understanding to other oscillation phenomena, REM theta? Yes, absolutely. So this we can find spindle-like peaks in all stages of sleep and also during wake and resting state that we've looked at. So yeah, it's absolutely applicable. Okay, let's see. This is for single EEG use? I assume that means single channel. Yeah, so I think one of the work, so is this just applicable for single channels? So some of the work that we're doing is actually expanding this into not just a two-dimensional thing, but actually in a spatial dimension. So you have time, frequency, depth of sleep, amplitude, and spatial location. So that's some of the work that we're trying to use to try to expand this. Okay, and I'm going to just answer one more question so that we can get on to our final speaker and we can answer the rest later. How do you account for changes in aperiodic activity to find the TF peaks? So we actually compute an estimate, sort of non-parametric estimate of the background aperiodic activity, which we remove before we do the peak detection. So that sort of allows us to adjust that. Again, there are more sophisticated ways of doing this, but we find that this works very well.